Great, it's 11:03. Let's go ahead and get started. Hello and welcome everyone to the 5th event in our MetroFlow Corporate Member Launch Seminar Series. I'm Christina DeCoste, Director of the Flow Cytometry Resource Facility at Princeton University and President of MetroFlow, the New York, New Jersey Flow Cytometry Users Group. Since we weren't able to meet in person in 2020, the 15 members of the MetroFlow Steering Committee created this launch seminar series as a vehicle for our corporate members to reach our MetroFlow audience via this virtual platform. This session is the 5th, and there will be several to follow through 2021. You can find our previous online events archived on our YouTube page that you can link to through our website, metroflow.org. As a disclaimer, MetroFlow remains neutral and in no way endorses any one vendor or their offerings over another. All corporate members have the opportunity to submit a proposal to present in this launch seminar series. And the purpose of these events is to showcase the innovations in the field that we can all take advantage of to better support our scientific and research endeavors. Well, we hope that all of you will be able to take what you learned from the seminar series back to your labs and that this information will be helpful in your future endeavors. For some housekeeping on the Zoom platform, I'm sure we don't need any of this anymore. We've all done enough of these, but if you select the Fit to Screen window at the top, it's best viewing, and please use the Q&A button at the bottom of the screen to submit questions, which will be read to the speakers at the end of the presentation. So please feel free to submit your Q&A anytime during the presentation. Please use the chat function to report any technical difficulties. And before we get started, we'd like to thank today's speaker, as well as you, our audience, for your time and participation. With that, I'd like to introduce our speaker, Dr. Klaus Herrick of Chromotek, part of Proteintech Group, who will be telling us about their nano-secondaries.
Thank you very much for your kind introduction, Christina.
Okay, you can go ahead and share your screen.
Yeah, that would be nice. So
there you go. We can see it.
Does it work? Perfect.
Yes.
Yeah. So warm welcome from my side as well. I'm presenting out of Germany, so it's a very unique setting. And I like to talk today about so-called nanobodies that we have developed for next generation capture and detection of immunoglobulins. And we call these nanobodies dependent on application, either nano-secondaries or nanotraps. So let me find a pencil. So the content, so the talk is structured that I will first introduce nanobodies. What are these? Then I will talk about nano-secondaries for immunofluorescence, nano-secondaries for flow cytometry, where we have a limited amount of data, and talk about the Proteintech antibodies for flow cytometry. So what's a nanobody? What's a nanosecondary? So we are all familiar with the concept of a traditional antibody consists of a heavy chain and a light chain with the hinge region. As it appears, llamas, like other camelid animals, like dromedaries, camelids, llamas, and such like, they do have these traditional antibodies, but they also possess a second type of antibodies, the so-called heavy chain antibodies. And when you compare these, you realize they have an Fc portion, and they're called heavy chain antibodies. So it's a very descriptive name. They don't have the light chain, they only have a heavy chain, but that's not the entire truth. They also lack the CH1 domain of the traditional IgGs. So what's been called IgG3 among the alpacas, it's a heavy chain antibody, and due to the fact that it does not have the light chain and the CH1 domain, it's just 100 kilodaltons, whereas a traditional antibody is about 150, 160 kilodaltons. Now, the binding domains of these heavy chain antibodies, that's what we are interested in, that's what we are developing. that what we are generating by immunization of alpacas, that's a nanobody. A nanobody is just 15 kilodalton in size and the smallest antibody that is available. We do precisely label these nanobodies with, generally speaking, 2 fluorophores. And if we have developed a nanobody against an IgG, in this case, that's a rabbit IgG, we are offering these nanobodies, labeled nanobodies as secondaries in a mixture that is specifically binding to rabbit IgG, the Fc domain, as well as the Fab domain. A total of 4 nanobodies is binding, carrying a total of 8 Alexa fluors. So you can do a very reproducible, very quantitative labeling of your antibodies. Validation and quality of secondary antibodies is becoming more and more important. In the past, we had the discussions going on about the reproducibility, availability, and this OEMing and trading of antibodies, mainly primary antibodies, and also different campaigns were started, like this one headed by Matthias Uhlen, with that validation proposal of these 5 pillars for antibody validation. But that also runs into the area of secondary antibodies. And I'm showing here a quote of CiteAB. It's from citeab.com, from their website. It's a blog that Rebecca Sadler has been written, where she's really encouraging scientists to properly cite the secondaries. But it should not stop with a properly citation. It also should comprise a proper validation of the secondary antibodies. And that's what we believe is something that our nanosecondaries do very well. So when you compare a traditional polyclonal secondary antibody labeling of a primary, so here we have the primary antibody that binds to the epitope, here in purple, and then we have this very arbitrary complex of polyclonal secondary antibodies labeled with a large number of fluorophores all over the place. Now compared with a nanosecondary, like on the previous slide, or 2 slides previously, you have the epitope and you have the binding antibody to the epitope, and then you have a very precise, very defined labeling with the nanosecondaries. And here we have, for instance, a much smaller label to epitope displacement. So the resolution, image resolution, is much higher than comparing the label to epitope displacement in these dimensions on that hand side. So rather than having a very arbitrary complex, we're having a very defined complex that we can use to detect and quantitate antibodies. The nanobodies, the nanosecondaries, are monovalent. And that's a very nice feature, because now you can do a one-step incubation, which means you can incubate your primary antibody with the secondary antibody, with our nanosecondary, and apply it to microscopy. In principle, in a standard workflow, you first have to apply your primary, do the washing, apply your secondary antibody, do the washing before you can actually start doing the microscopy. So it reduces hands-on time, and it's obviously much more gentle for your sample. One-step incubation, just to show some examples. So here we have a mouse IgG1 primary and a mouse IgG2b primary here. And on the left-hand side, you can see the one-step labeling. On the right-hand side, you can see the sequential labeling that we've done in parallel. Basically, you can't spot a difference. It works very well. High resolution, I mentioned that. The nanobodies are very small, just 15 kilodaltons. And this is an image that customers of us have been done from the Netherlands. And they have been looking into Vimentin and Hela cells using a special Leica high-resolution microscope. And this bar here indicates 1,000 nanometers, a micrometer. And detection has been done using an anti-Vimentin mouse IgG1 primary antibody and an anti-mouse IgG1 nano-secondary. And it's a really beautiful resolution showing the power of nano-secondaries and high resolution. But higher resolution does also work on other samples, like here shown by a customer of us from Germany on mouse kidney cells, in fact. So here we have a mouse kidney slide. And that slide has been probed with an primary antibody against a cellular protein, and using a secondary polyclonal secondary antibody for detection compared to a nanosecondary, as shown here on this cartoon that you probably will remember. And by just applying the nanosecondaries, they gained a resolution increase of 20 nanometers. It doesn't sound a lot, but in the world of super resolution microscopy, 20 nanometers is quite an improvement. And our customer, Florian Siegeres, was really excited about getting these data and happily to forward these data to us. High resolution in comparison to confocal. Here we have a laminar staining. In confocal, you can imagine it's actually 2 layers of the lamina, but when you utilize a STAT technology afterwards, you can see that it's actually 3 layers of the lamina perfectly stained with an anti-rabbit IgG, anti-lamin, and an anti-rabbit nanosecondary for staining. Also, tissue penetration is something that is improved by using small nanosecondaries. So here we have customer data about showing Drosophila larva brain. The brain has been probed with a primary antibody and the counter stain for DOPA for the cell cycle, cell nucleus. And what you can see here is the red signal is the nanosecondary that is being, the red signal is being equally distributing throughout the larva's brain tissue. Whereas here on the right-hand side, where a donkey anti-mouse secondary antibody has been used, a conventional nanosecondary, you can see that the red signals are only on the surface of this tissue. So here clearly indicating that the nanosecondary penetrates better into the tissue. I frequently get the question why this makes such of a difference if you use a conventional primary antibody. I don't have a good explanation, but the data really speak for themselves. Cross-reactivity is something, and background and multiplexing. So what we've done here is we have used our nanosecondaries and applied them to subclass samples that were dotted on a membrane, either mouse IgG1, mouse IgG2A, 2B, C, mouse IgG3, and rabbit IgG, and we have probed them with the corresponding nanosecondaries, and you can see no cross-reactivity at all, neither from rabbit to any of the mouse subclasses, but also within the mouse subclasses, there is no crosstalk. We've compared that with a pre-absorbed goat anti-rabbit IgG antibody, and you can see that even though it's pre-absorbed, you still get some background towards other species and subclasses. So with the nanosecondaries, you have no cross-reactivity to IgGs of sera and sera, in fact, from other commonly used species and subclasses. They have a very low background. They are recombinantly manufactured, so they're very reproducible and they have an unlimited supply. And you can use these for multiplexing. Multiplexing, just to show you a number of examples. So here we've shown a multiplexing of 2 times mouse and 1 times rabbit primary antibody. So the yellow is a rabbit anti-lamin, then stained with a nanosecondary. The green is a mouse anti-IgG, anti-COX4. And the magenta is a mouse IgG2b antitubulin, primary and incubated and stained with a nanosecondary alpaca anti-mouse IgG2b. And then you can see the overlay, a perfectly nice picture, very detailed and very impressive in terms of cross-reactivity. But cross-reactivity does also work on 3 mouse primary antibodies. So here we have Hela cells that are stained with a mouse IgG2b anti-Lamina, a mouse IgG3 anti-MOT, and a mouse IgG1 anti-Vimentin primary, all with corresponding nanosecondaries. And also here, as there is no crosstalk, you can do a perfectly well cross multiplexing of that sample. Now a customer went a step ahead, and he in fact applied the same subclass mouse IgG primary antibody. So he has used a mouse IgG1 antitubulin antibody and pre-incubated it with anti-mouse IgG1 alexafluor 88. And he then mixed another mixture of a mouse IgG1, so the same species and subclass, anti-LAMP1 primary, mixed and pre-incubated with the nanosecondary against mouse IgG1 alexafluor 568. And here we have the single channels after application to the sample, and here we have the overlay. So that's very, very, very interesting. It probably still needs some optimization towards a possible crosstalk, but in principle, that's really a great step ahead for fast imaging and possibly also fast flow cytometry. Now, what's our portfolio? Our portfolio is we do have an anti-rabbit that is also co-reactive to anti-human. We are working as we speak on an anti-human, solely anti-human IgG nanosecondary. We also have mouse IgG1, mouse IgG2b, and mouse IgG3. And what you can see here is where these actually bind. So the rabbit is a mixture of 2 nanobodies targeting the Fc domain as well as the Fab area of the primary rabbit antibody. The mouse IgG1 is a mixture of 2 Fc-specific nanobodies. Same holds true for the anti-mouse IgG2b. The mouse IgG3, which is the least abundant subclass in IgG in mouse, is labeled with an Fc-specific. And we thought in terms of signal intensity, we add a 3rd fluorophore to the nanobodies. In terms of adding signal, it is 2 point something, as quenching starts significantly when you add a 3rd fluorophore to it. As these nanobodies are rather small, they are lots of drama in terms of labeling, as the fluorophores, not the most hydrophilic chemicals at all, really do not increase the solubility of the nanobodies, and we have lots of precipitation during production. So that's why we have 3 fluorophores with a degree of labeling 3 for the mouse IgG3 nanosecondaries and 2 each for the others. As I mentioned earlier, we do have limited data on flow cytometry available. We just have purchased a flow cytometer, and these are one of the first data that we have generated. And this is Hela cells that are stained with a rabbit anti-human CD55. And what you can see here in red is Hela at unstained. The grayish is Hela and the anti-rabbit nanosecondary. And the blue one is a signal of the Hela cells with the rabbit primary anti-CD55 primary antibody, as well as the nanosecondaries. And you can see sufficient log steps in between to clearly differentiate the two signals. Not as clearly differentiated, but still very well differentiated here on that Jurkat cells that are either CD3 positive or CD3 negative, as probed with an anti-CD3 mouse IgG1 and Nano-Secondary. Quick introduction of Proteintech. Proteintech is a German company that was acquired last October by Proteintech out of Illinois in Chicago. And the Proteintech group comprises 3 product ranges. It's a Proteintech antibodies, where the impressive antibody portfolio against 13,000 human proteins is available. They are all in-house manufactured. They're not OEM'd. Also humankind, recombinant proteins, cytokines, and growth factors. They are manufactured under GMP grade and very unique. They are expressed in hex cells for native folding, for native maturation, and native human glycosylation. So you get human proteins expressed in human cells for real human behavior. And finally, our Chromotek brand, we are around since 2008, and our portfolio is based on nanobodies that we are incorporating into reagents for elevated assay performance. Quick introduction into Proteintech's antibodies. So from that 13,000 human targets, 2,500 targets are already validated by knockdown or knockout. All antibodies are validated for fit for purpose application dependent on the target. There is, I believe, now more than 110,000 product citations. The majority of the products are rabbit polyclonal, but there is an increasing number of mouse monoclonals, about 2,000, I believe. What's also contributing to the antibody value continuum is rabbit recombinant monoclonals, chimeric antibodies, lots of specific ELISAs, but also authenticain premium ELISA kits, nucleokine monoclonal antibodies, correlate for flow cytometry, and classical clones for flow cytometry that you probably know by far better than I do. Just to show a little bit further detail on the classic clone portfolio for flow cytometry of Proteintech. These are popular CD and extracellular markers from legacy clones. There are about 160 unconjugated, about 250 conjugated to FITS, PE, and APC, but also to what we call correlates, which are, let's call them derivatives of Alexa fluors. We also have 42 isotype controls, and there are more and more colors coming soon. The flow cytometry validated portfolio comprises about 900 antibodies, 140 correlate conjugates, and these are correlate 488, correlate 594, and correlate 647 for multiplexing, and also many intracellular targets. And flow cytometry is very important for the product portfolios development at Proteintech, and lots of effort and focus is given for the expansion of antibodies that can be used for flow cytometry. So it's probably worth a look to check the website once in a while. What are correlate antibodies? Correlate antibodies are synthetic fluorescent dyes. They're equivalent to Alexa fluor dyes in terms of brightness, stability, and spectral profile. And we do conjugate new targets to correlate as we speak. The antibody validation for flow cytometry is very important because it's very close to our heart. It's really scientists working for scientists, from our bench to your bench is our phrase here. And we do have an award-winning validation for our protein binding affinity and target selectivity. we, in terms of flow cytometry, we check signal separation with IgG isotope controls, and only if the signal is 3 times of that control, it's passed. We also carefully titrate the optimal antibody amount and product-specific protocols. So really antibody where antibody are provided and to give the optimal experimental details in terms of cell type, antibody amount, fixation, and permeabilization method if required. Why choose Proteintech products, whether these are antibodies or nanobodies, for flow cytometry? Because of our high-level extensive in-house validation, our extensive target coverage. We have an individual tailored technical support that we're happy to provide. We have product-specific protocols, antibody by antibody. We have next-day delivery, competitive pricing, and a comprehensive antibody guarantee. We don't ask, we just replace the antibody if it doesn't work for you. With that being said, back to my green background of Chromotek. Thank you very much for your attention. Questions are welcome. And as we are in America, no presentation without disclaimers. Thank you very much.
Thank you so much, Klaus. Hey, everybody, I'm Thomas Miller. I'm on the steering committee at MetroFlow. Just want to remind everybody that you can ask questions in the Q&A, and I'll relate those to Klaus. I can start off with one question. So you've began testing this on flow cytometry. You said you've recently got your cytometer installed. Have you noticed any issues or anything that, any special technique that we need to worry about when staining with this, or does it totally work with the one-step
one-step incubation? Yes, dilution 1 to 500, I believe, if I remember correctly, and very straightforward. There is basically no difference. They're easy to apply.
But that's interesting you bring up the dilution too, because that was another question of mine is, do you have to worry about the nanobodies being taken up by unbound primary antibody, since we're using that in excess?
We can't exclude, because when we determine the off and the on rate, as we know the affinities of these, they are in the single-digit nanomolar or sub-nanomolar range. So over the course of an hour or so, you may see some off rates and some leaking. That could actually happen. And here, obviously, the monovalence of the nanobody is a disadvantage because they don't gain from avidity that you have on a standard bivalent secondary antibody. But the leaking is rather low. It may cause trouble if you have, for instance, if you co-stain a sample with 10 times, 100 times, and 1,000 times. So you have and all the same isotype and species primary, then you may get up with a mixing. But if you use different species and subclasses, there is no problem. The now secondary will dissociate and disappear basically. But in our hands, within the normal time period of experiments, we haven't seen that.
Okay. Lydia has a question. Is there a need for blocking against non-specific binding?
I missed that.
Is there a need for blocking against non-specific binding?
Yeah, in immunofluorescence, we do block. We use BSA. There is no special alpaca serum or something like this is needed. So BSA is perfectly fine. Okay.
Thanks, Lydia. Vera has a question. Is there interest to validate these nanobodies for use in small particle flow cytometry, i.e. extracellular vesicles, viruses?
If you want to try, please contact Fran. We're happy to provide some samples.
Okay, great. And then Ira has a question. Do you have any quantitative comparison between intensity of labeling with traditional and nano-secondary
We have some customer data that are looking pretty good, but we don't have the experimental setup to really do this photo counting microscopy. So nothing that I can share, and nothing that would satisfy a really independent peer review right now. But at some point, some customers will do that. It's beyond our laboratory instrumentation capabilities, unfortunately.
Thanks, Ira, sorry. Vera has another question. Do you use specific protocols to remove the unbound fluorophores?
No, it's simple wash steps. It's when what you could do is you could calculate the molar ratio, but if you want to do that or you wash it away, doesn't really matter.
Okay. And Lydia, our last ask, can you share these slides? Maybe she can reach out to Fran.
Yeah. I PDFs them, or Fran has them already as PDFs, so that's no problem at all. It will be our pleasure.
Great. Let me see. I don't think we have any more questions. I'd just like to remind everybody that this also will be recorded, and we will post it on our YouTube channel. And you can find that YouTube channel by going to metroflow.org. Thanks, Klaus, for a great presentation.
Thank you very much for your attention and wish you a great day.
Thanks, you too.
Bye.